might
Maybe
make
work?
it work.
Oh my god!
Well, because you
And then I did one. I think like I did a few without it, and then I did with, and it did both the same issue. So,
Yeah, it's really tough to track down where the contaminant was,
I
but
went through like made the whole grow back thing and with the oat and it's like a hit and miss situation.
Oh, did someone come in?
oh no, I just saw that you have the YouTube up.
Oh yeah, I got it working.
Yours looks so good.
Oh well, this I purchased.
Oh man, I was like, man, I was like, yes. I was like, woo.
Although mine tends to look good when I make it. This is made with some homemade stuff. I just brought this as an example. It was a spawn that I made almost a full year ago. That had just been sitting on a shelf, and it's going to still work. It's just going to take a while. But I find for growing mushrooms, oyster mushrooms at home, I try to just go with pre-made spawn because
Better.
well, you can choose strains that are proven. You know, like really nice. Hi, how's it going?
Good, how are you?
Oh, pretty well. Because you can use strains that are developed for commercial production, and so, yeah. Well, I'm gonna wait to start doing the actual cultivation stuff until we get everybody here. But in the meantime, I figure we can just do a little Q&A. I brought some mushrooms. If anyone wants to look at mushrooms, and if anyone else brought mushrooms, I can help you ID them. As far as this stuff goes, it'd be really hard to well, it's really hard to troubleshoot what exactly happened. I see it looks like
do
this.
you like okay? When
Go
you
ahead.
apply liquid starter, I've seen a few different techniques. Like, do you do the whole burning the needle, or do you just wipe it down with high alcohol base?
Oh, I usually sterilize the needle. Yeah, I have like a
How do
little
you
torch.
Okay. Is it better to torch it than to just wipe it down with the high alcohol rate?
Definitely. Because
Okay. Maybe that could be it. Because
if y'all want chairs, by the way, I can bring over some
Yeah,
chairs.
there's a ton of them over here.
I didn't do that.
Yeah. So the thing is, what alcohol does is it mostly cleans dirt off the surface, and then it'll kill a lot of different bacteria and some viruses. But what it won't kill are bacterial spores or fungal spores. And so that's kind of the thing with using alcohol is it's not really.
No, you're good.
Okay.
Want to give a share for yourself?
Yeah, we'll be moving around a little bit. But so, what was the bait? What did you use to grow?
That's like what is it called? Brown rice something? It's like it's now. I bought it online. I think it's brown rice. Yeah.
Yeah. I see there's some mica in here, or is it?
I think whatever they had put together. I know. I was like, that doesn't look like just brown rice, but yeah.
Okay, so for making your own spawn, all you need is you can use a couple different types of grains. So the commercial stuff is done on millet, and I highly recommend millet.
Millet, okay.
Yeah. The reason being that
It's better than oat.
So it breaks up into much smaller pieces. So if you see in the bag that I made over there,
This one here.
yeah, you see how small those little dots are? Those are all inoculation points. So if you're using a larger grain, really when you're inoculating your bulk substrate, you want to be spread out as much as possible because it doesn't matter how much mycelium is in that grain. It just takes like one or two cells to be an inoculation point, and more so matters how much surface area you're covering, and that'll lead to quicker colonization and more complete colonization. So millet's an ideal one for oyster mushroom spawn, and you can buy that at a feed store. I think pearl millet might be one of the names for it, and it's super easy to work with.
Is it better to just buy, just so to try it, like just mill it from the store?
Oh yeah, you
Okay,
go to
okay.
go to the bulk aisle of the supermarket if they carry it. And I don't have the recipe off the top of my head, but I feel like it's 50-50 ratio. Do you remember?
I think it is. A quick Google search will give you a range.
Of what?
Yeah, because like there's a lot of cultivation sites that people will recommend, but I think it is 50% of the weight. Or yeah, I can't remember. It's been a couple of years since I've made grain, but or made
Spawn.
thank you.
Yeah, and then basically what you can do is, if you're using jars like these, did you make these yourself?
No.
Okay. And were the jars already filled with the material?
Yeah.
Okay, so
I've tried making my own and have not been successful.
fair. I mean, it's tricky.
Dang.
Um,
So what would be like? Is it because I bought liquid starter to inoculate, and then I also wanted to use the liquid because I've been also growing them with liquid starter.
okay.
Keep growing more, so I don't have to buy the same. You know.
Yeah.
So I have
feel it.
like some of that, like the oyster mushroom. I have portobello and like a few. I have it in like, like they have a sugar water thing.
Yeah, of course. So it has like an airlock on it and everything. It's well sealed.
I have changed it to something like that.
Yeah.
I mean, it didn't go bad
Okay.
with it.
I mean, liquid culture or liquid inoculant, I usually stray away from it. It's, in my experience, it's more of an advanced level thing because it gets contaminated extremely easily,
Oh.
and it's very hard to tell when it's contaminated. Even if it looks like it's clean, there could be yeast or bacteria growing in there, and you wouldn't know until you transfer it to a substrate, and then you get contamination. There can also be spores floating around in it, waiting to germinate once it hits the right medium. So liquid culture is the most efficient way, and it's the cheapest way.
What's your way?
So usually what I do is I just go from grain to grain, and I do agar as well. And so agar is for me the best way because you can always see how clean your material is.
Do you want to take us over to the whiteboard and the YouTube, and we could actually use kind of like go over the entire process because this is a really good simplified version of the process.
I'm so excited.
You want to?
Sure. Yeah, I don't think anyone's on the channel, but it's being recorded. So.
Okay.
Yeah, this is actually it's a great little
Ooh, the floor plan.
visual aid of
Love it.
all the different ways you can cultivate. And so the method that you're doing is the liquid culture,
Yeah.
and you're starting with these syringes, right? You bought the syringe.
I bought the syringe.
Okay. And so what you want to do is grain jar, and then you try to inoculate some sugar water for fresh. Yeah, so that's I consider that more of an advanced level technique because it's so hard. It's so easy to contaminate, and it's really hard to tell once you contaminate. The most foolproof method, in my opinion, is to do agar, and that requires some sterile working conditions. Do you have a still air box that you use?
No.
Okay,
No.
so that's the easiest move, and
Oh, really?
I don't have one that I can.
A still air box.
Okay, so it's as simple as buying one of these containers.
Okay.
Just a clear plastic tub.
Okay.
And
And then creating a hole in gloves.
don't even have to do the holes.
No?
I did that for years, and then I had a friend who'd been doing it without the holes, and it works just fine.
No. So how do you keep it sterile?
So what the still air box is doing is it's preventing the rain of spores and bacteria from falling down onto what you're working on.
Okay.
Which means that everything underneath it has to be clean. So you have to start with a really nice surface that's like a hard material. I mean, the prep is not ideal, but it's, you have a metal table or you have a plastic table, and you wipe that down with bleach, and then let the bleach,
Dry.
dry. then what you would want to do is you take some books or something to prop up the container. And I wish I had an example of something. These are all filled with stuff, but here, I can actually one of these. So, what you want to do, and of course this is going to be a clear plastic container. And then you would want to prop it up on. Yeah. We're just going to pretend that these are something to prop it up on. And so with your clean surface, you would
like that.
prop it up like that with enough space to reach underneath.
Oh,
And
for
then
your
reach
hand.
underneath. Mhm.
So you clean it up completely, clean the table.
Yeah, and so the main objective of this is most of your contamination is going to come from gravity and from wind currents blowing around. If you're on a really clean table, your hands are clean, the inside of the container is clean, the air that flows around is not going to pick up any bacteria or spores that bring it in there. It's not foolproof. You're not going to get 100% success. The only way to get 100% success is with one of those. And it's not like those are completely out of reach. I bought one used for a couple $100, but again, that's not quite this nice. It's more of a smaller device. But this will get you really far. So my friend Damon and I, we went on an expedition to Arizona over the summer, and we brought a still air box and some other materials to do mushroom cultivation, some pre-poured agar plates and stuff, and we were able to cultivate a bunch of different mushrooms that we found in the wild on agar in a hotel room, just by sanitizing the surface.
Love it.
The thing that's gonna make you most successful is gonna be how you move. So even without a still air box, you can get clean cultures. You just have to move quickly and move precisely. Because if you leave the dish open for too long, it's just a game of math. It's like how many spores are floating by at a given time. So I've definitely done culturing where I didn't have a still air box at all, and it just was really quick, open and close, and
Doing the agar.
So you can also pour your agar dishes in a still air box. It's a little tricky because it tends to fill up with steam as you're pouring. You can also buy pre-poured agar dishes, which I don't recommend because they're generally pretty expensive. But
Well, agar is easy to access in the Asian store.
oh yeah. If you're gonna pour it yourself, yeah. And you don't need to buy petri dishes. A lot of folks use little deli cups, like the things that ketchup comes in.
Oh,
little
yes.
ketchup
I know what you
cups.
mean, yeah.
And as long as when they're in their sleeve, their plastic sleeve, they're generally pretty sterile. So as long as you open them inside your still air box and pour in there, and I'm gonna hold a workshop on this whole thing. This is something I have planned.
So how does that help, like the agar thing? So how do you go from agar to what? You go from agar to that?
Yeah, so super easily. So in your still air box, so what you would do is you make jars like that, and it's pretty easy to do. So you would get a jar, you would get a lid, and you can buy these little like vent things from Micropose, the company that sells different mycology supplies. So you would buy the jars, drill a hole in the lid, put the sticker on it. You would put your millet and water in the jar, and then if you have an Instant Pot at home, you can sterilize them in your Instant Pot. Then once they're cool, you can go into your still air box, unscrew the top carefully, and take a little piece of agar that's been grown with the mushroom tissue, put it in there, and close it, and then you have grain swap.
Wow.
And when you're using agar, it's much easier to tell if you're running into contamination. Because
it'll
it's
show signs of it.
Yeah. It's growing on a two-dimensional medium. Oftentimes bacteria is just, it might make the water turbid. It might be cloudy when there's bacteria, but sometimes it's not. I've run into issues. I've bought liquid culture from reputable suppliers, taken it, and in my laminar flow hood or my biosafety cabinet, I've put it on an agar dish and seen contamination.
Whoa.
Because it's that easy to get contamination. When I make liquid culture at home, I do it 100% in one of those, and I have it on an orbital shaker in there, and in really high quality lab conditions, and that's how I get it 100% free of contamination.
So I'm going to put in a plug here. I'm an officer with Counterculture Labs. So if you become a member of Counterculture Labs, you have access to all this equipment, including that, and you have a sterilizer here, and you have all the things you need to come and do this. If you're interested in doing it in an existing lab setup. But yeah, just taking divvying up the agar plate and putting it straight into a sterilized grain, like he was saying, millet or rye berry. As long as that's at the right moisture content, and as long as you don't have contamination, that still air box transfer is gonna fully colonize, and then you can go, like he was saying, from grain to grain like that, or you can just go straight to a grow phase to make a bag. And what's the proportion of moisture to grain in the jar there that you were pointing out? I'd have to remember. I mean, it's been a couple of years since I've done made grain or sawdust spawn, but ultimately each medium. So millet I think is 50. It's like half. 50, 50. I can't remember which, but yeah, a quick Google search will tell you what medium you want on a mycology website. Somebody will say what worked for them, and I think for us, we use the same content. But the moisture content needs to be right, because if it's too heavy, it'll get anaerobic, and you'll get that green stuff.
Yeah. When I made my oak one, when I was looking online, they said to put gypsum to help, because I feel like I do put it, and I still have issue both in the jar and both in the grow bag. Maybe because I put.
Well, it could have been a liquid culture contagion. So if it really.
Hello, here for the mushroom meetup? Yep. Oh, awesome. What's your name? DH. Oh, no worries. I wasn't going to get started doing the actual stuff until about 5:45, because I know people need to find parking and traffic and all that stuff. So, we're just having a quick discussion about cultivation. Some folks are asking some questions, so feel free to join in.
And there's this visual here if you're interested. Yeah, this is something I would definitely take a picture of and kind of mull it over in your head, because at any part of the grow cycle, it's good to be familiar with. If you got contamination here, well, was it your liquid culture, or was it where? It's good to know the whole cycle of.
Yeah, and I think there's enough interest that the next workshop I do will be entirely agar.
Yeah,
just
totally.
Agar in a still air box, agar in a flow hood, agar every which possible way.
Yeah, I think that's a great idea.
Because that's what got me started. I got very frustrated using liquid culture. I had mixed results. Sometimes it worked, sometimes I had contamination.
I didn't know it was going to be that hard, because I had been trying for a whole year. And my husband knows my struggle.
Yeah, it's a lot of work to fail, but if you do it right, you can have success right out the gate.
Oh man.
Probably.
Because I don't know, for me, the reason why I didn't do the agar thing is because I didn't feel like I had that still air box or clean air enough that I had that lab feeling to do it. That's why. Because when I see those things on YouTube, it is very sterile looking, and I'm like, I don't think I even have that in a little apartment.
Yeah, I mean, it's one of those things where it seems like it's there's two sides to the coin. It's really simple, and it's also really hard. It seems like it's more challenging than it is, but it also can be extremely challenging. So experience solves all of those. So once you start getting comfortable with it, and you start to see, you're going to have some trial and error. You're going to have to see what causes failure and what causes success. But you're very likely to have more success than failure if you just follow some simple conditions. But just having agar is makes life so much easier because you can really see what's happening.
I think I'm gonna
Liquid culture, who knows what's growing in it? And even as I said, even from reputable suppliers, I put it on a plate and had yeast grow with the mushroom because in the dirt the mushroom's living with all these organisms and it just doesn't care. So it'll happily live in liquid culture with bacteria and yeast and you won't even know. And these days I really don't. The only thing I do liquid culture for is for doing high molecular weight DNA extraction.
Oh, wow.
I don't use it for cultivation just because, well, one, I'm not growing enough mushrooms for it to be that big of a deal. I feel like if you're a commercial grower, if you're growing like hundreds of pounds of mushrooms, there's no other way to do it because you're going to waste your time with grain. But if you're just doing it for you and in your home, like it's too much of a headache, and you need too many you need too many things to go right for it to work out for you. Do you plan on doing like a mushroom farm or anything?
No. I just want to grow like a ton of, oyster mushroom, maybe portobello, just to have and to eat.
Mm-hmm.
Yeah.
Eat and eat.
Well, oyster mushrooms are the easiest mushrooms to grow. Once you get past the struggle that you're having, once you have it looking like this, then there's very little chance of failure after that point. And so today the workshop we're going to do is the lowest tech, the simplest, easiest way to do it, which is 99% effective. In fact, I've never had it contaminated on me. Of course, today is going to be the day because I said that. But we're taking advantage of two important things, and that is that oyster mushrooms deal with contamination extremely well once they're established. Once the mycelium is established, it'll fight a lot of things off. In the wild, it eats bacteria. It's a bacteriophagic. It's also nematophagic, so it also eats nematodes. It lives in a really terrible environment. It lives in these like decaying logs where there's just so many other organisms competing for that habitat. So it's pretty good at fending off contamination. The other advantage that we have is that we're using a substrate that has virtually no nutrition whatsoever. We're going to use hardwood sawdust pellets, and there is almost nothing in there. It is just lignin and cellulose.
What type of hardwood?
I use oak.
Okay, because oak is the best.
Oak is the best, and it's also, I think you can use pretty much anything with oysters.
I don't know.
It just can't be.
Maybe eucalyptus or pine.
Yeah.
Those are
Anything that's really heavy in aromatics, those compounds are made by those trees to prevent them from getting infected with fungi. So, you see a pine log that's fallen. There's only a few mushrooms that you'll ever see growing out of it, and those are really well adapted. So, like Gymnopilus, that has whatever enzyme it takes to break down those heavy aromatics. But oyster mushrooms, you only rarely see them on conifers. When you do, it's cool. You should definitely keep them. I have a few cultures of oysters that I found growing on fir trees and pine trees. And so whether that's a different species or whether that's just a different, just has a little bit different genetic software built into it so it can handle those. Hardwood, mixed hardwood, cherry, fruit wood, almond wood, pretty much anything you can find that isn't a conifer and isn't eucalyptus.
Yeah, and you can take that agar, like he was saying. You could. What he did was he cut off a piece of oyster from a Douglas fir up north, and he gave it to me, and then we ended up making some bags out of it, and it was some of the most prolific oysters I had that season.
Yeah, it was a grand fir, but still, nonetheless, a true fir is actually even harder to grow on than Douglas firs. Yeah.
So you said that if you're growing commercially, you use liquid culture.
Yeah, and that's just for the sake of efficiency because you can set up a system where you have these sort of rubber gaskets on your jars or bags that are self-healing. So you can just take a needle with the liquid culture, jab it in there, inject a little bit, and then move on. Whereas if you're dealing with mixing grain with grain or mixing agar, there's more motion. You have to cut a piece of agar out and then move it in physically, or you have to take some grain and move it and then shake it around. If you're just injecting, it's like super fast. So all the commercial mushroom farms inoculate using liquid culture.
So they're inoculating these, what I see as army of grain jars. Is that what they are?
So yeah, you'd have like a lot of volume of liquid culture, and you would hit each one of those. But in like a home environment, you're going to have to heat the needle every time you come out. You're going to have to, whereas in here, you sterilize. I think you would still do that, right? Yeah, you'd still cook the needle in between each injection, but that's a lot easier than taking a slice out of the agar and doing all those extra steps, is what he was saying.
And commercially, you'd be using bags. You would never use grain jars. I personally have gotten away from grain jars. It's a little bit less environmentally friendly to use plastic bags. I buy plastic bags that have some sort of process where they break down easier. I'm not sure if that's good because I think it just breaks down into microplastics, but it's going to do that anyway. Jars are just kind of a pain in the butt because they're solid, and so when
They
you,
take up space.
well, yeah, and when you're trying to break up your grain,
We can't. You have
like,
to take it out.
yeah, what you do is if you have a mountain bike and your tires are pumped up really, really high, you take the jar and you just bang it against the tire until the grain breaks up
But you can't do
enough. But you know, commercial operations are always going to use these single-use plastics, which is you can reuse these. You actually can,
Yeah.
depending on how you're using them. I have clips, and I didn't bring any with me today, but it's like basically a clip that seals it.
Yeah.
Creates an airtight seal. So you could, if you wanted to be a little bit more environmentally sensitive, you could reuse this 3 or 4 times. There's a certain point at which it'll start to degrade because you're subjecting these to, you know, pressure cooking. Pressure cooking is essential. You have to hit 121 degrees Fahrenheit, 121 degrees Celsius, or 250 degrees Fahrenheit. I don't know how these things work.
I have to look at my pressure cooker.
Anyway, it's above the boiling point of water, and you have to hit it for a certain amount of time, and that's to kill bacterial endospores. Like Bacillus is a genus of bacteria that's in everything. It's inside your body. It's in the soil. It's all over your grains that you're using, and the endospores can survive incredible things. pH changes, heat changes, so you have to keep it. That's why pressure cooking is required. Otherwise, the bacteria will grow faster than the mushrooms.
How do the mushrooms get out of the bag?
Out of the jar. Or out of the bag is easy. You break up the stuff, and you kind of break it up with your hand and then dump it. With the jar, as I said, you have to bang it against something to break up the grains, and then you pour it out.
So the final result will come after you've mixed your grain into the growing medium. So what he was talking about earlier was the wood, the oak, the hardwood that he has in that bag is mixed amongst that grain culture that's in the big white plastic bag. That plastic, so like all the little millet that he was talking about upon like spreading out, all of this is millet. So each one of these little, so that's like, that bag could make a couple hundred of these maybe? I don't know.
This is 20 pounds. And 20 pounds can make, at a 10% spawn rate, that's 200 pounds of bulk substrate, right? If I can do math.
I think so.
What? I guess I didn't ask my question right. Are the mushrooms strong enough to puncture through the plastic?
No, you have to at that point. So once he just put those millet in that bag, and he'll get to this later.
Okay.
But once the mycelium has run throughout that bag, you can puncture holes, or you could puncture them now, but you run the risk of contamination. But he'll get to that.
Okay.
Yeah, we're not gonna poke any holes today. I like to let it go for a little bit. Mushrooms are aerobic organisms for the most part, but they can survive on very little oxygen. So you could probably let it colonize the whole thing without poking a hole, and it'll do its thing. I'm not sure how it manages to deal with such little oxygen. I don't even have vents on most of my
Is it
liquid
because of the
culture.
plastic? The plastic,
Oh yeah, there is probably some
Hydrogen
confusion through there.
peroxide
right?
that's mixed
Because
with the
when
wood.
you like
Uh,
have
you can add. I can answer that.
Yeah, but with my liquid culture jars, I don't actually have vents.
Oh.
I leave a little bit of airspace and it's fine. They don't need enough. They don't need that much oxygen. And I mean, my agar dishes, they're wrapped in parafilm and in plastic bags, and those sit inside boxes, and those cultures live, for a year or more. There is some gas exchange, but it's very little.
So, since I don't know if my liquid containers of starters are free of contaminant, so it would be ideal to create like an agar thing, squeeze some in it as in a sterile environment as possible, and then let it see it grow, and let's see how it grows, and then go from there.
Yep,
Okay.
absolutely. Yeah, if you're doing it in a still air box, your success rate is not going to be 100%, but it's going to be pretty good, and you'll be able to see where there's contamination pretty easily. So, yeah, and I will. The next workshop we do here, I'll do agar because it's a great skill, and it doesn't require fancy equipment. Just an instant pot, and if you can get away with just a microwave, but it's all depends on how successful you want to be. If you want 100% success, you'll have an autoclave and a laminar flow cabinet. If you want to be 90% successful, then you'll have an instant pot and laminar flow. If you want to be 80% successful, then you'll have instant pot and a still air box. So it all depends on what your tolerance for failure is. It's very rewarding once you figure it out.
Okay. I hope
I stuck with it, and now I have 80 different species of mushrooms now in my library at home.
Oh, my
I-
goodness.
Yeah.
Can you get spore out of dehydrated one or no?
Yeah, as long as it's dehydrated at low heat.
Oh, I don't know. I'm just saying because his dad gave me some dehydrated mushrooms. Technically to cook, but I don't know.
Okay, well, if they're dehydrated below like 115 degrees Fahrenheit, then you probably have a chance.
Really?
Yeah. But if it's dehydrated above that temperature, then it'll kill everything. So, but think about when you go hiking in the canyon here and you see oyster mushrooms growing out of a log after the first rain. That mycelium has been sitting in that log for the whole dry season. So where I used to live in Castro Valley, it would get 90 degrees every day, 100 degrees every day in these areas, and the log would be completely desiccated, like no sign of any moisture in it. And then, a week after the rain, there comes your oyster mushrooms. So they can be extremely good at surviving, at going into some sort of stasis. And the way that I preserve my cultures at home is I take little circles of the agar and I put them into sterile water and screw the cap on, and they'll live for years. Just sitting in distilled sterile water.
Oh, really?
With no nutrients, no air, no oxygen at all, completely sealed off from oxygen, and they will survive, room temperature.
Wow.
Another way you can preserve mushroom cultures is using a freeze dryer. You can freeze dry your mushroom cultures, and they will just sit there completely freeze dried, and then you put them back on agar, and they come back to life.
Wow.
It's crazy. They're pretty good at surviving harsh conditions.
Need to know.
Yeah, but to answer that question, those holes were put at the end of the grow cycle so that the fruiting body would leave. But that bag that you see there how did you mix that today or?
Yes.
Yeah. So like in a week from now, that should white should spread throughout and then once you feel that the mycelium has infiltrated that sawdust bag, then you poke holes or
Yeah, just use a push pin and poke, go around it. They make these, it's like a ravioli cutter or something. Someone was talking about it on a YouTube video where it's like a wheel that has little like,
Just punches holes.
yeah, so you just run it up and down the sides and it'll punch tiny little holes.
And the mushrooms can grow out of
Oh, I
holes
know
that
what
small?
yeah, the mushroom will be breathing at that point. Once it starts to make the fruiting body, it takes air and precipitation to keep expounding how big the mushroom will get. It'll as it evaporates, it kind of helps the fruiting body get bigger.
They'll fruit through the holes. The hydrostatic pressure of the mushrooms expanding will push the plastic out of the way, or the hyphae will grow through, the mycelium will grow through the tiny hole and then fruit outside of the bag. So I grow a lot of lion's mane at home, and I'm not always very good about transferring it to this bulk substrate on time. So the grain spawn that I have in these bags, these holes here are like 0.3 micron or something. The mycelium will actually grow through the tiny holes in this filter patch, and I'll get a mushroom fruiting on the outside of that. So yeah, they'll make it through tiny holes. They seem to figure it out. The best thing you can do is pack these bags really tight. And I just made this in a hurry just so I could bring an example of what one kind of looks like, and this is a small one. But when we make them, which will be soon, hopefully some more people show up, you'll want to pack it in really, really tight, because what tends to happen is if there's a little bit of airspace, they'll start to fruit underneath the plastic, and it's not impossible to fix that. You just kind of make a little hole for them, and they'll figure it out. Oh, did it disconnected?
I don't know.
Okay. We're still there.
Something, anybody, why haven't people been able to grow truffles?
Truffles, I mean, I think you can grow one particular type of truffle.
Yeah, white truffles.
But other ones, there's a lot of mushrooms don't want to create a fruiting body under sterile conditions. A lot of them don't like the sterile conditions because a lot of them need the symbiotic relationships with the ecosystem that they prefer.
Yeah, there's a lot of things. So black truffles, Perigord truffles, have been cultivated in Sonoma County. And they used to be cultivated in France. Before World War II, there were tons of truffle orchards. But then World War II happened, and those orchards got abandoned or bombed or destroyed. And it's like a 30 to 50 year investment of time to get truffles to grow. White Alba truffles have never been able to be grown outside of certain parts of Italy and I think like Albania and that whole area. And they exist only with host trees, so they get all of their nutrition from the tree. Well, most of their nutrition comes directly from the tree. So if you don't have that right tree, they're not going to grow. The other nutrition they get is from the soil and from the microbes. So they need this crazy complicated environment where you have the right tree, the right microbes in the soil, the right soil composition, and then maybe they'll grow. But again, it's one of those things where you can inoculate a truffle orchard and you won't know if you're successful for 30 years. So a lot of people just don't want to put that kind of work in. But someone in Sonoma County has done it with black Perigord truffles from France. Those are the ones that go for, $800 a pound. Not as expensive as the white truffles, which will go for $2,000 a pound, but still pretty good. Well, maybe I'll give it a couple more minutes. Full 10 people registered, so Plus a couple other folks that wanted to join. Oh yeah, we can look at the mushrooms.
I got me like a starter thing, like you know, like I said, yeah, and I expanded on like this, coastal fire, one plant, and put in like this, use the one that was already technically grown out, and then I just put it into this, fiber substrate, and I told my daughter, and then it did grow. But I couldn't. So I-
Yeah, it's actually pretty cool here.
But it's like the most amazing thing when you get going. Oh.
Have you been out hunting much?
It's a little. You grow,
Okay.
cut it, grow it.
Rachel?
Yes.
Okay,
Yeah.
it's been a long
It's
time.
That we get shares instead? Yeah.
Yeah, if you'd like, I did bring some mushrooms for show and tell. I don't know if anyone else did. So we can go over them. Y'all have cut mushrooms in the wild?
No, I don't know how to. Would you like this shirt? Let me eat this shirt. Oh, yeah. So you know how to forage.
Oh yeah, that's what got me in the mushrooms.
I'm so jealous that you can forage because I find that like scary.
Oh, it shouldn't be scary. It should be so much fun.
So, no, so want the right one.
Well, yeah, I mean, it's tricky. So these, at least 2 of these are edible. At least 3 of these are edible. At least one of them is poisonous.
Right.
But it's all about getting comfortable with groups. So maybe you won't be able to identify something to species, but you can get a good idea of things to avoid. So this right here is Swillus pungens. And it's called that because it has a somewhat pungent smell. I think it actually smells really nice. This grows with pine trees. You'll find it with Monterey pine and bishop pine and a couple of other different pines in our area. It's edible. People are split on whether it's a good edible. If you talk to people from Eastern Europe, it's a good edible. If you talk to people from the United States, it's not a good edible. So there's a big cultural
Is it
basis.
flavor or what do you call it?
Texture. So when you cook this up, it's kind of squishy. It has kind of a squishy, slimy texture. Not a lot of people like it. This has pores on the underside. So all these other ones have gills, except for that. This is a gilled mushroom, and this one has pores. Mushrooms with pores, almost all of them are edible, to some degree. Only 3 of them in California I know to be poisonous. And there's maybe 1,000 species that have pores. So if there's only 3 of them, 0.03% chance. And the ones that are toxic are pretty easy to identify. So,
Are they like more colorful?
oh, definitely, yeah. Here, let me see if I can bring them up.
They're like, don't.
There's some around.
Yeah, of course. So this book,
Wow.
critical. If you're going to be hunting mushrooms in California, you need this book, Mushrooms of the Redwood Coast.
Wow.
So there's a whole section on all the pored mushrooms, and I'm going to flip to the page that has the poisonous ones. So here's the 2 main poisonous ones in California. They're both in the same genus, Rubroboletus, and you'll notice that the pores on the bottom are red.
Oh, yeah.
The only poisonous ones have red pores. So if it doesn't have red pores, but it has pores, and it's a bolete, because there are mushrooms with pores that are not like this. There's like shelf fungi, like have you heard of like reishi?
Yes.
Reishi also has pores, and it's not poisonous, but it's not really edible. It's not something you wouldn't take a reishi mushroom and like cook it up for dinner because it's so hard. You break your teeth trying to eat it, but you can make tea out of it. So any sort of typical mushroom like this that has pores on the underside, you know that's a good place to start. Because it's very easy to recognize. There's a lot of features that you can compare with. And so a lot of people's first experience hunting mushrooms is you go for porcini mushrooms.
Yes.
And those are unmistakable. They're these big, chunky, beautiful, stately mushrooms that are highly regarded edibles. The other one that people tend to go for, and I don't have any examples here, but that's really easy to identify, are chanterelles. There are a couple of lookalikes for chanterelles, and you have one that's poisonous, and it's called Omphaliotis olivescens. But I wouldn't say it looks that close. You'd have to be very careless to just eat mushrooms without being 100% sure of your identity. So I can show you pictures of both of them and see if you tell me if they look the same.
I would say maybe the color is pretty close, but the body definitely will take on a different feature. But the color is what lets people see the orange, and they're like, "Oh, chanterelles!" But then it turns out it's the jack-o'-lantern mushroom, and it makes you pretty sick.
Am I wrong to say that I'm seeing more like availability of mushrooms, more and more, like in supermarkets and even more variety?
Oh, you're not wrong.
Yeah,
Like, in the Asian store, of course, I'm like, they
Well,
seem
yeah.
to have it anyway, but it
Well,
seems to have even more variety now. Like,
yeah. I
was
mean,
like, whoa.
at Safeway 2 years ago, oyster mushrooms were $30 a pound. Now they're $15 or $6 a pound, depending on the week. But yeah, you're right. Because they're pretty easy, but supply and demand, ultimately, they are producing more. There are more mushroom farms now, but there's never enough mushroom farms either. So,
All right. So
it's room to grow.
here are two of the different species of chanterelle that grow in California.
So that's the one that's edible, right?
Yes, edible and highly desirable. And then this is the one that people think looks like.
That's not sweet.
See the color? It's very different.
They're darker.
And these one, if you look close, they don't have real gills. They've got ridges, whereas this one has true gills. And there's an easy way I can mark the pages, and then I can pass it around so you don't lose them.
So in other words, you just have to, if you can afford, bring the book with you.
The best thing to do, here, I can pass it around, the pages are marked. The best thing you can do is bring the mushrooms home with you
Oh, and then go and cruise.
and sit with the book. It's very hard to bring that into the field. You're not going to fit that
I
in
was
your
gonna say
pocket.
it's kind of a.
Yeah, you're not going to stop and take it out of your backpack and it's raining and no. Hunt the mushrooms, pick ones that have pores, pick ones that look like chanterelles, bring them home, sit with the book, and also you can have people help you. So I know a lot of folks don't like Facebook, and I am one of them. I don't like Facebook, but for the mushroom community, it is the place where everybody goes because the community is so spread out. They need to have a place.
To
You know,
just meet.
Exactly, because it's like if you go to a bar and you start to talk to somebody about mushrooms. Nowadays, you might get lucky, but 5 years ago, no one's gonna have a conversation with you. They're gonna be like, "What's wrong with you? Why are you talking about this weird shit?" So that's why everybody sort of got on Facebook. There's a lot of Facebook groups. One is called the California Mushroom Identification Forum. And it's a Facebook group where you can take pictures. If you take good pictures, you don't want to just take a picture of the top, right? Because can you identify a person just by looking at the top of their head? Exactly. So you want to take a good picture of the mushroom from the top, from the side, and then you post it to that forum, and people can identify it. Or you can get an app for your phone called iNaturalist. And I usually have a talk that I do, but I didn't bring the cable to connect my computer to this. But I can post some slides to the chat after this. And it has all that information of the books. Yeah. But iNaturalist is also an incredible resource.
Oh, the app, right?
Yeah.
Oh, yeah.
It has a computer algorithm that will help identify mushrooms, but it's not 100% accurate.
Are there any Discord communities?
There probably are. There's Reddit communities, although I don't recommend the Reddit community. I'm not part of any specific Discord. But with iNaturalist, I can show you just how it works. So let's say you have a picture on your phone of a mushroom that you took. So you would just push this button, you would choose an image, and here's one that I took today. And you would select it, and then you would press this button. What did you see? So at the top here, and there it's going to go, and it's going to use a computer algorithm to try to identify it for you. Now the mushroom I took a picture of is Kibaloma. The first suggestion it gives me is Kibaloma. It does a very good job at getting you in the ballpark. I wouldn't trust my life on it because it's just an AI. It's just looking at it's just comparing images, and sometimes I'll take a picture of something and it'll tell me the absolute most incorrect answer. But it's a good start. But then I can select it, and I'll press this checkbox, and it uploads it, and then it goes to this website where a community of people are going to be looking at these. So let me scroll down and find one. Let's look at somebody ID this. So here's one that I took a picture of called Gymnopus fuscopurpureus. And I uploaded it like that, and then this guy Kyle, he came along and he saw that and he confirmed my identification.
Oh, you're doing that.
And Kyle's not an expert. He's my friend, and he knows that I know the mushrooms, so he's agreeing with me. But if you don't have your friends that agree with you all the time on there, then you'll have somebody who knows a lot about mushrooms, like my friend Alan, will come on there, and Alan generally identifies most of the mushrooms in California.
Alan Rockefeller is his name. In fact, here's one right here. He identified for me that 12 others, the honey mushroom I showed you earlier.
So if you arm yourself with the right tools, foraging is not scary at all.
Yeah.
And even if you do make a mistake, there's only 3 or 4, maybe 5 species of mushrooms in California that can kill you. So a good thing to do is learn what those look like. And if you learn what the deadly poisonous ones look like, then you've eliminated the chance of dying. If you eat this, you won't die, but you will get very sick. So it's not to say that you should just go eat random mushrooms. I brought these truffles too. These are undescribed species of truffle in the genus Melanogaster.
So truffle is part of the fungi?
Truffles are fungi.
Oh.
These don't have a super strong smell. They did, when they get really old, they smell really intense. And that's part of the truffle life cycle. So these are related to these. These two are more closely related than this is to this. And that's just because of convergent evolution. So the same truffles that you would buy at a Michelin star restaurant are not even, they're as related to this as you. So it's just that things to choose a different form or shape. And just like evolution decided that this should have pores and that these two mushrooms should have gills, even though these two mushrooms are not even remotely related, they're very, very distant. But they came to the same conclusion that gills were a good idea. Surface area. In this case, pores is the surface area. The actual spores are produced on the inside of those pores, in those tubes where the spores are produced and they fall out. And the gills are produced on the side of the gills. In this, the spores are produced in these little chambers called gleba. And the reason why a mushroom would take on this shape is because it's trying to avoid the spores getting destroyed before they're mature. So if this starts to fruit and then it gets really dry and windy out, and before the spores have a chance to form, it's going to dry out the mushroom and the spores are never going to mature. And it's never going to accomplish the task of spreading the spores. Or let's say this fruits and you get a bunch of insects and they start eating the spores. Spores have oil in them, and oil is a rich source of food. So the insects will eat all the spores before they have a chance to mature. The mushroom doesn't succeed in spreading. But if you grow underground and then all your spores are inside, surrounded by this membrane that keeps insects out. If it's really dry outside, it doesn't matter because this is underground. So it's going to maintain optimal conditions for the spores to develop underground. When this is mature, when the spores are ready, what it's going to do is it's going to release a scent, an aroma. And that aroma is going to
So
attract.
that's why it attracts like pigs to.
Yeah, so that smell, the smell of culinary truffles is a male pig sex pheromone. So it is smelling like there's a smell in there that's the same as a male pig who really wants to make babies. So the female pig is going to detect that and dig up the truffle because it gets the pig gets very excited. He thinks that babies are going to get made. So much the same with this. This would attract maybe a squirrel. This is an undescribed species. Nobody knows its ecology or anything. So who knows what this is going to attract. It definitely does a good job of attracting curious mycologists. So maybe it evolved to attract us. But
Are there any above ground signs?
yeah, so this one actually fruits mostly above ground. I brought it as an example, but like it was, let's say this was like the duck, it was poking out like that. And actually a lot of truffles will do that. They'll poke out just a little bit. Some of them will be completely underground.
You know, I would really like, since you are so expert mode, like, I would love to have to do it. And we go.
I know.
That'd be so fun.
I
Just
want to
like, no, like, it'll be called a hike, but with the intent of foraging and looking.
yeah, I think I want to do that. Maybe I'll do that not next week, but the week after. Next week
Okay,
is
because it's still
really big
raining.
week for me.
Still raining, right?
Well, I'm doing a huge event next weekend in Sonoma County. I'll be doing a 3-day workshop doing PCR and sequencing and stuff.
Wow.
What is
Yeah.
that?
Chain reaction.
Yeah, like amplifying DNA and then using the DNA to identify the mushrooms.
Yeah, we
So-
do that in our lab. for example.
Yeah.
We have PCR.
Oh, I learned to do that here, and I think Rachel came to a couple workshops when I was just getting it figured out. I didn't know what I was doing. I screwed it up so many times,
It's not easy.
and now that's like my I do that for a organization called the Fungal Diversity Survey. I take care of all their sequencing.
Kidding.
Well, I don't necessarily do all of it, but I manage it. So for certain species that people mail it to my house, and I'll do PCR on it, and then for other ones.
So I mean, is it like DNA sequencing or what is it?
Yeah.
Oh,
So
really?
it's barcoding, so you're looking at a short piece of DNA and using that very short piece of DNA as a barcode, as like a fingerprint.
Okay.
So if you see that barcode again, you can say, oh, this species of mushroom.
So you're doing workshops like that. I know Alan Rockefeller does workshops like that. The guy who was mentioned earlier.
Wow.
And he does it actually.
That sounds fancy.
He has his own lab set up and he also
Is
does.
there a meetup link for that activity?
What's that?
Is there a meetup link for your PCR demo?
So this is going to be at Soma Camp, which is it's a private event that costs $600.
Oh, boy.
Um,
Like, stuff.
So it's through the Sonoma County Mycological Association. It's an entire weekend where everyone camps out in the woods, and that's just
That
one.
sounds
That's
fun.
one of many workshops. There's workshops where people are
Fun.
doing forays. People are using mushrooms to dye clothing. I mean, it's everything mushrooms in one place.
That's
Wow.
next weekend.
Yep, that's next weekend, and it usually gets booked up really quickly. But a lot of people are canceling because of the weather. It's going to be miserable. It's going to be cold and
right.
rainy. We might not have electricity. So a lot of people are canceling. So you might be able to get in
Okay.
if you want to spend $600 to spend the weekend in the cold, freezing.
It's okay.
There's also a mushroom festival in Marin next weekend as well. So that's, I don't think
Is there a,
it's not going to be as good. Maybe soon. Where's that going to be in Marin? Yeah, I don't know where in Marin, but I know that. I mean, Soma is like the Super Bowl of mushrooms on the West Coast.
Yeah, Super Bowl is a good term.
Yeah, because it's like, that's where you're going to see like a lot of people who are on the forefront of mycological stuff. And a lot of people are in this area too.
Yeah,
I mean, Alan is a huge.
Yeah, Alan Rockefeller is a very generous person with his knowledge and time, and taught me to do a lot of these molecular techniques. And I was an idiot, I still am, but at least I know how to do PCR. But yeah, thanks to him, and that actually inspired me to go and get a degree in molecular biology. So yeah, got into it that much. So he's a great person to follow. Again, he does a lot of stuff on Facebook because it's just the nexus of the community. So, but you can just add him as a friend and he'll accept it, and you can. If you post mushroom pictures on the forums, he'll come and occasionally he'll identify things. But he's like a cornerstone of the community here in the Bay Area. Well, I'm not sure if we're gonna have any other folks showing up, so I'd love to get started making some mushroom bags. I brought enough for about 15 people, so I don't see 15 people here. So it looks like we're all gonna have to go home with a lot of mushroom bags.
You just made her day.
I love it.
Cool.
Makes me happy.
So I'm gonna put these aside, and I'm gonna wipe everything down again because these have probably been spreading spores and potentially contaminating our work area. Not that I think it's gonna be that much of a problem.
Do you want to have both these tables for packing? Just one table?
Yeah, I feel like we can put completed bags on this table. But we should probably just wipe it down a little bit.
My previous job, I
Yeah. Yeah.
This is fun.
Oh, yeah, totally. They're kind of a pain to clean, but
And then we went with the stars.
yeah, you'll definitely want to, when cleaning those out, you want to wear a mask and try not to breathe any of that green stuff.
I will.
Yeah, cleaning those jars is the pain in the butt that he was talking about, and also those lids. Unfortunately, they're not as reusable either because they'll rust, and then that becomes a contamination point.
Yeah, so there's a website, MicroPose. You should put it in your phone, MicroPose, MicroPose. They sell some equipment that you can use for cultivation. They have little stickers that are that have injection ports. They have stickers that are allow air, and they also sell like different sort of cultivation stuff. And then the other website I get equipment from is called shroomsupply.com.
That's where I got.
That website's great. They
And
have
that's where I got those.
Okay,
And
cool.
like some other stuff that I have not opened because I'm still waiting for it to get there.
Yeah.
How high the alcohol do you need to have in there?
70%.
Okay.
You don't want it any higher than that because it's not very effective at higher than 70.
Oh, really?
Between 60 and 70 is ideal
Okay.
because alcohol will evaporate so quickly that if it's like 90% alcohol, then it will evaporate too fast.
Oh, okay. I thought the higher the better. I'm wrong.
Well, you need some water there too. That it's the interaction of the water, the alcohol, and then the cell membrane. So all three of this, you have to have the water in order for the alcohol to interact with the cell membrane. Enough. Micropose with two P's. Micro P-P-O-S-E. Take out my fancy watch.
It's a chickpea, I didn't know that.
I missed that.
No, I don't know if I like it, but
Yeah, it's.
it's
I've had a high tech watch from like.
I will look
Yeah.
Oh, yeah.
Thank
that-
you.
No, I need
Mushroom,
that.
it'll-
That I have to start getting healthy, so like it helps track, all those things.
But, no, I just wanted to check out.
COVID was hard on me.
I feel you, bro.
It's totally fine.
I did
A couple years of
We're
sitting
gonna
home
make
drinking
one.
beer,
That's
like,
why
it's not good for you. Okay.
I can't wait for all of
So I'm gonna just set up a little station here. So this will be the bagging area, and let me just wipe this whole
Do
thing.
we need gloves or are we good?
Everyone's just gonna wear gloves.
Oh,
I can't tell you exactly, but I would say between 3 and 4 weeks is typical. It all depends on how much of the spawn you use. And since, yeah, we're probably gonna go with over 10%, just because I don't need to stretch it out. And the higher ratio you go, the better
Oh,
yield.
yeah.
With sawdust. Sawdust has no nitrogen, almost no nitrogen. And mushrooms need nitrogen to fruit. So that's the caveat to this low-tech method. And I'll do a quick introduction. So the method we're gonna use is just using hardwood sawdust pellets, water, and spawn. Those are the only ingredients. Hardwood sawdust pellets are available at a bunch of different suppliers. I got mine from a store called it's called the Chef's Store. It used to be called Cash and Carry. It's like a food service supply
Yeah.
store, and they had them for super cheap. I think it was like $11 for a 20-pound bag. And a 20-pound bag will yield 50 pounds of substrate, and 50 pounds of substrate should get you at least 20 pounds of mushrooms. So there are, yes.
I think there's some on the side there.
Yep, so there's gloves over here. I brought these big clunky gloves. Where's that spray? Oh.
Stuff in the white stuff in the bag
It's,
is
it's,
spawn.
it's a spawn one. So we're gonna start off with sawdust and all. I wanna put a little bit of that in, and it'll help us grow.
So what is that stuff?
Oh, he bought it.
Spawn.
I don't recommend making your own spawn until you have, the right setup to do so, because it's just you're gonna suffer when you fail.
Oh.
How much is this bag from Amazon?
From Amazon, Mino bought it, but it's not that expensive.
Those are
I would say it was like $100.
There's a medium over there too.
There's mediums over there too.
And it went right.
So we're not following this.
No, that's
We're more in the
a
bottom.
general
We're just-
explanation of the
we're
process.
more the bottom. We're gonna,
Yeah,
we're the latter part.
exactly. The last two steps you see on that
That's what we're doing.
is this is
Yeah.
this is like hay or
Excited.
sawdust, and we're adding the grain to it, and then eventually at home you could poke holes, or you could poke holes now, but it makes more sense to.
So the grain is the spawn.
So yes, exactly. So this is millet. The grain in here is millet, but so it's grain spawn. You can also make spawn with sawdust. And essentially, what he's doing right now, by having made this can colonize, and then you could add the sawdust to a different medium, but it's not going to be as effective as if you spawn it with grain. But you can make spawn with sawdust, because all like what Hart was saying was, all you need to do is take a little bit of this genetic material into the right environment, and it will spread. But it just needs to have an edge on the environment and be able to take over faster than anything else there.
So when you buy spawn, is that the small quantity they sell it?
No, they have smaller. They have
Yeah.
smaller bags.
Yeah, at Amacel, this is the smallest quantity they sell. Amacel provides mushroom spawn to commercial farmers. So that's like, we buy from them because it's the cheapest.
The volume, right?
Yeah. So I'm gonna go ahead and weigh this out while I talk. And again, this alcohol is not saving the world here. It's just killing whatever bacteria are on the surface that are alive. It will not protect you from total contamination. So I have 14 pounds of mixed substrate here.
Do you ever use peroxide as well?
No. I mean, I've heard about that. So that's 1 pound 9 ounces.
Drawn on the board, the peroxide.
some people use it, but you know, everyone's got their trick. So what this is those hardwood sawdust pellets, and all I did was mix them with cold tap water. That's it. And I let them sit. This actually I mixed last night, but the other 130 or so pounds that I made up right before I went. And hardwood sawdust pellets are by themselves completely sterile. To make the pellets, you take sawdust and they put it in a press, which increases the pressure to the point where no living organism can survive. Yeah, heat and pressure that's involved. And so you're starting out with something that's completely sterile. You're adding tap water, which is not sterile. But the stuff that's in the tap water that you have to be concerned about is mostly bacteria. And bacteria are really bad at eating cellulose and lignin. Bacteria need nitrogen. This substrate has almost no nitrogen whatsoever.
So you don't need like hot water and.
Nope, you don't need hot water or anything.
Wow.
It makes life so much easier. Your nitrogen source is going to be the nitrogen that's in the grain soil, at least at first. When you're done and you put this in your garden, then it's going to start getting nitrogen from bacteria and other things. And you'll get more flushes. But right now, it actually feels warm from.
Wow.
It actually feels warm from all the metabolism that's going on.
If I touch it?
Did you spray your hands by chance?
Oh, goodness.
Here, here.
Spray me.
What's the percent of water? Like,
Oh, great question. So it's 60% hydration. So how I do that math is just I start with 20-pound bags of the substrate, and if you do 20 divided by 0.4, that'll give you what the whole amount is, which is 50. And then if you do 50 times 0.6, that gives you how much water. So it's really easy with a 20-pound bag because then it's 20 pounds of substrate to 30 pounds of water. And it's better if you can convert it to kilograms because then you can easily figure out how much water to put. I have this postal scale here and I just weighed out 1 container of water and I made a mark of when it was 5 pounds and then just filled it up to that mark. It doesn't have to be exact. But too much water and you'll go anaerobic and you'll end up growing bacteria instead of mushrooms. And then too little water and you won't get mushrooms grown. The mycelium will go all throughout the substrate, but it won't have enough water to actually produce mushrooms.
are there any nutrients that come in when there's hot water since it's not distilled water, but there's dissolved stuff?
Oh, for sure, yeah.
So there might even be the advantage of using it's not as distilled if you're using sawdust.
Absolutely, and I would never recommend using distilled water unless you're trying to do like science experiments, because the mushrooms are pretty. They're not going to get harmed by the chlorine in the water, and there's minerals, and minerals are a good thing. If you use distilled water, there's no minerals. How's it going?
Good. I'm sorry I'm late. I'm Andrew.
Don't be sorry. You're all good. You're all good. Andrew, my name's Hart. I'm conducting the class, and I've got Tommy here. Tommy's helping applied mycology. Culture labs. Have you been here before?
I've been to the Omnis, but the counterculture lab. So.
Cool. Well, welcome. We were just talking about mushrooms for about an hour or so, and we just started doing the cultivation part, so you're not really missing anything.
Yeah, sure. There's a lot of good stuff. I'm glad to hear.
Cool. So what we're doing here is I have this substrate, which is hardwood sawdust pellets that are hydrated with cold tap water. And then I purchased mushroom spawn from a commercial website, and that's what that stuff is there. It's grown on millet. And what we're going to do here is just mix all this stuff together. It's very low tech. Anyone can do it. You don't need any special equipment or sterile conditions. I just recommend a little bit of isopropyl alcohol. And all of us are going to get to go home with a bunch of bags so we can grow mushrooms. So, I'm busy talking and I haven't been doing anything. I've just been mixing this. So come a little closer if you'd like, so you can see what's going on. So that mushroom spawn is grown on millet. You can see it's all dispersed here through the hydrated sawdust pellets. And that mushroom spawn is what's going to colonize this substrate. It's also what's going to provide nitrogen. So what I was saying is the reason why we could do this really low tech without any sterilization is because the hardwood sawdust pellets, they come pre-sterilized. When they're pressed into the pellet shape, it kills everything in them, and they also have no nitrogen, or almost no nitrogen. And bacteria need nitrogen to grow. Your main contaminant that you're going to have to worry about is bacteria. Because bacteria grows way faster than mushrooms could ever possibly grow. So if you take the risk of bacterial contamination out of it, then you can do this really low tech without any sterilization, and you'll probably be fine. Oyster mushrooms are also really good at withstanding contamination.
Yeah.
So once you've mixed all this stuff together, then I got this roll of poly tubing. Again, this is single-use plastic, and there's other ways to do this. I don't feel great about using single-use plastic, but this was given to me. So it's not like I'm gonna save the world by not using it. Then you just get these regular old zip ties that you can get at the hardware store. People might look at you weird if you walk out of the hardware store with plastic sheeting and zip ties, but you know.
This semester, I'll try to make you one. I'm trying to make a ceramic version of a grow kit, like an external part, so you can easily open it up and clean it, and one that would fit in the Presto, or maybe an Instapot. That would make more sense, right?
You seal these with food safer.
Just zip tie.
It's better, it seals it tighter.
It-
All right, so then I'm gonna decide how big I want my mushroom bag, and we can be generous because not a lot of people showed up. I have my scissors here.
I think it's up to the other official with the gloves. I'm standing so far away.
No, you're
So-
fine.
This amount is I weighed this out to be 6 pounds. And I was like, oh, everyone will go with home with a 6 pound bag, and now that I'm looking at it, that looks so small.
I know.
So we can make bigger bags because I have 130 pounds of material. So so maybe we can make something more like this size. And then everything's been sprayed with isopropanol, isopropyl alcohol. And then I'm just going to grab one of these scoops and fill it up.
Do you notice any difference in colors? It seems to have how much soap you put in the bag. If there's a lot of internal density or heat that's generated. Does it colonize differently if it's a small bag versus a large bag? Food wise or?
I feel like small bags tend to colonize faster, but I don't know if that's like just because it looks like they do. You're asking a hard questions.
Right.
No, it's okay. I did test amount of spawn used, and I estimated this to be about 10% here by weight. It's not exactly. I think I put 1 pound 9 ounces, and this is 14 pounds of substrate. So I don't know how that math works out to be, but it's around 10%.
Yeah, the more spawn you use to your growing medium, like the sawdust, like 10% is going to be twice as fast as 5%.
Oh yeah, you can fit a lot in here.
No
And you're-
kidding.
The more spawn you use, the more the faster it's going to infiltrate the medium and fruit and go to fruit faster. So some big productions, they'll use really high volumes of spawn to grow mushrooms faster and to turn over the yield faster, but it's just a safer bet to go with more than less, because
Really, like.
you want to be able to outcompete any contaminant. Because like right now, the sawdust is getting contaminated by stuff, probably me, but
But yeah,
Cool. So once I've put what I feel like is enough, this is probably like 12 pounds. To make something this size, and this will yield probably 4 pounds of mushrooms over the course of its lifespan. The main thing that mushrooms are made out of is water. So once you get one flush, you might get a second flush, but if you don't, what you can do is you can soak it. And we'll talk about that in a bit. So I have another bus tub here. Thomas, would you mind just spraying that out, and then we can kind of get started. I got 2 scoops. I brought these 2 to stir things up if you want to, and then we can just kind of get the process going, and I'll just supervise and ask questions or answer questions and stuff like that. But I'm trying to press as much of the air out of this as possible. And the reason being that if there's air pockets, the mushrooms will try to fruit in those little air pockets. And they'll kind of be stuck underneath the plastic. So you'll have to free them. Yeah. And then we can bring over, so if 2 people want to grab one of those tubs and bring it over here, the top one I think weighs like 70 pounds.
Okay.
Where?
Oh, we can put it on the floor.
Oh, floor? Oh, okay.
somebody has the,
Tell me your name again?
Andrew.
Andrew. Andrew. Yeah.
All right. All right.
A
So,
little bit, like up and
so it's probably a good idea to go ahead and
spray again.
yeah, remove the.
I
alcohol.
definitely might renew.
And just try to get all the nooks and crannies of the glove, because it's all going to touch the stuff, and you just want to get as much of an advantage for the mycelium to take over, not anything else.
So maybe I'll move this pretty close to the table. I'm just trying to avoid getting too much sawdust on the floor. I know it's going to happen. So what we can do is grab the metal scoop and
Can't be too careful, right? Oh. I was like, okay.
we go ahead and tear this out, and then start scooping some stuff in here, and then we'll weigh it out and figure out how much spawn we should put with it.
Right there. Open it.
And you don't have to be extremely sterile. We're just eliminating as many possible sources of contamination. Nothing here is sterile. This is, you could probably get away with not spraying yourself and not spraying anything. You'd probably still be fine, but.
Right, pass it down.
So is this how the pellets heat?
So the pellets come in dry in a bag, and the reason why I hydrated them ahead of time is because it takes a little while for them to absorb. So I give it about 2 hours to soak.
Yeah, I'm not going to give up.
Oh.
I'm just testing.
2 hours?
Yeah, about 2 hours to soak in completely. And I'm just gonna stir this
Look
up.
at that.
So
That's the
this
same
is just
container that I have
soaked
home.
in syrup?
Oh, I'll wait for the, for
This
this
is just
first
soaked in syrup.
That's it, just water and the hardwood sawdust pellets. Make some or any of those other things that people say you need to add.
Yeah, you won't need the bag just for a little bit. Put it in here. Here, I'll show you what you missed.
Okay, thank you.
Yeah. So what he has right there, that white bag, is essentially what exactly.
We did the all do.
Range with mycelium.
What size bag is
He
it
added
added
to
to?
10
pounds, 12 pounds?
Is there
Whatever.
different size?
Just depends on like the space you have and how big of a bag you're gonna make yourself. That one looks like probably 10 to 12 pounds.
Yeah.
So 7.
And so what's the melon? mycelium infiltrates the whole bag. It will
I think it's fine
start
there.
to
Is it manual?
no, it's done. It's
It's like 10 pounds, 5.8 ounces.
You probably hit the whole button on the time. Maybe because it's like.
ultimately, we got to this by one layer of it, the way
So then we can start filling with the rest of this, and then we'll just cycle it over. So then for this, you want to put, for 10 pounds, you want to put about 1 pound of spawn. You can be generous with it though. And I wish I brought something to weigh the spawn out on. Here, I have an idea.
Awesome, thank you.
There's like a paper plate or something.
Oh, I have one? Oh, okay, I was like, let me make sure everyone have one. Okay, so.
So it's a 10-to-1 ratio.
Yeah, approximately. It's a very hand wavy thing.
It's hand wavy, but like, what side do you want to err on? Like, do you want like the ratio to be like more material than spawn, or do you want more spawn? Well, like pretty much I'm saying like, is it better the ratio greater than 10-to-1, or is it better less than 10-to-1?
I would say more than 10 to 1 because this substrate is very low in nitrogen. And so it's the energy, the nitrogen that it's going to get to make protein is going to come from the spawn itself. So it's kind of like you want more spawn. If you aim towards 15%, it'll colonize faster and you'll get a better yield. I'm going to try experimenting with using some nitrogen supplements, but I'm afraid of like having a bacterial disaster. So
I mean, what supplements are you thinking of using?
like urea,
Oh, wow. That is
or just homemade urea.
Oh.
My wife might think I'm a little weird for that, but coffee grinds are really prone to contamination. Perfect. There's something about coffee grounds that just invites trichoderma. I don't know what it is.
I think I tried doing with coffee ground and I would get the green.
Yeah, it's just something about coffee grounds.
Is that what's the
Well,
top of your?
that was like the first one a while back.
Yeah.
I would go through it and I heard about it and I tried it and it
Yeah.
just was a bad coffee.
I mean, I've had oysters just take off in my compost, just off the coffee. But every time I try to grow this coffee, I feel like when it's something about the sterilization.
Yeah, it makes it more prone to contamination. I don't know why. Coffee is really good in compost.
Yeah.
All right.
We're good. Yeah, so then we can just cut off, the appropriate amount of spawn and mix away. Just try to keep spraying your hands and spraying the tools as much as you can remember.
How much do you want?
So that's 10 pounds. So you can put a little over a pound, maybe like a pound and a few ounces of spawn in there. There is a knife over here, a serrated knife, so just be really careful.
I'll break it up.
That looks like, yeah, you can break it off, but I think it'll probably take more than that.
Yeah.
Is it a tear?
Yeah.
Wow.
Exactly, 1 pound.
Okay.
Yeah, you could probably go a little heavier than that. I mean, we're not going to run out of spawn. And after this, it's not like I have nothing to do with it after this. So
Yeah, spawn with compression reminds me of drywall.
But it's even more amazing though.
So, do you want to add?
so whoever wants to go next can add the rest of this to their bag, so we can start another container.
All right, I'll try and get some.
Or you can even just dump that into there and mix it all together. Maybe that's a better idea.
Okay.
Yeah, you
And
can
then
mix it in here.
Yep, and then we can start a new bin.
We'll move this one over.
I find restaurant supply stores have great equipment for this kind of stuff.
I agree.
Bus tubs, and these are ice machine scoops. And then I even brought another mixing bowl if we want, if we want to start moving things along, we can have another container.
If it's been out,
Oh, my pleasure.
yeah, I'll spread it. Just
If you want to start scooping into this, I'd do it, but then I'll be in your way. So.
Okay, we'll do it.
I'll start scooping.
And again, the numbers here are very hand wavy. Mushrooms don't do math, and so they don't really care about our math as long as we're in the ballpark.
Okay.
I think the fact that you already did the moisture thing, everything else is like, eh.
Exactly. The moisture is the most important.
Yeah.
If you're overhydrated, you're going to end up with a soggy, bacteria-filled mess.
Yeah, your grain is really in good, or the grain spawn's really.
So you said it was like a
Good content.
20-pound,
20-pound bag. It was like, I think, $11 at Cash and Carry.
but how much water do you put
30 pounds of water for 20 pounds of substrate.
in? Wow.
And if you have a scale that weighs kilograms, you'll make your life a lot easier.
Well, it's just 1.5 times as much weight.
Yeah.
It's a 2 to 3 ratio, right? 20 pounds to 30 pounds.
1.5.
Yeah, 1 point. Yeah, something like that.
Something like that.
Water.
60% hydration is what the
Okay, that
instructions
makes
told
sense.
me.
Okay. I was trying to remember the
Okay,
ratio.
yeah, this is like 10 point. No, it's like 10 pounds and something else. I was like 4.9, but
Close enough.
oh, 9.8.
Cool. And then you can go ahead and weigh out some spawn.
I mean, this is pretty good to go ahead and bag.
Okay. Yeah.
The knife here, you can cut off a piece.
Sure
Just be
thing.
careful and don't cut yourself.
Or,
Oh, sure.
so, what is the appropriate thickness?
Sure.
It's gonna be crumble anyway.
Yeah.
You'll be- Oh, I can't wait for all you guys to like just enjoy how it slowly become white like that, and then
Okay.
like
Then do I get?
slowly, like the whole
Yeah.
block, and then
Just have that. Just
slices, and then
The one rule is don't cut yourself or cut anybody else, and then we're winning.
Of course, do no harm.
I know.
No harm.
I
Then, I'm probably afraid on my hands again. Actually,
Thank you for coming
Right.
out. This makes my life
Thank you.
so much easier. Look at my phone, see if I more message. Yeah, I can't believe it. People paid for it, so.
So I'm so excited. You know what?
So does the alcohol have to completely dry before I touch the spawn, or? Okay. Gotcha.
Yeah, hydrogen peroxide and alcohol don't seem to cause contamination. They seem to just do the opposite.
Hey, guys.
I've never had any problems with rubbing alcohol.
Oh my
or
god,
hydrogen
I think
peroxide.
it'll work still.
But I've never really used hydrogen peroxide the way
Oh,
that
no,
most
I'm sorry.
cultivators.
I was thinking it's one something now.
I've never needed to use it. I generally, like, the less complicated I can make. That's been my goal. It's like, how can I dumb this down for myself so I don't have to think too hard?
Yeah.
Like, just try to avoid contamination from the get-go. I don't have to worry about it.
All right, here we go.
That's why I kind of want to be able to send a packet of this to someone so that they can make this on their own. You know what I mean? Like have like a mail order.
Right. Well, we'll go ahead and switch these out.
Okay, I didn't know I was going to do that much, but sure. And
So let's move this bus tub down, and then we'll get this one set up again.
okay, you can go and do that, and then.
Oh, wait, yeah, you got it.
You're welcome.
Okay. We'll break this up.
Yeah, right.
Ah, see, that's a good technique,
Oh, I
yeah.
love
you-
it.
The gravity smash.
That should be it.
You need a button.
Okay, when you're ready.
Most of what you want to try to evacuate is this right here. All the you do want to have a little bit of air in the inside here. This is fine, but like what he's trying to get you to evacuate is most of this.
Get this
this
done.
air.
Yeah, you want it densely packed, but it'll actually colonize faster with just a little bit of air in there.
Oh, so we don't
yeah.
need to.
What I would do is
Get all the
before
air
you put the tie
Yeah,
out,
you don't need to like
here's what I
compress
would
the
do.
medium.
I would go ahead
but-
and twist this real good, and kind of hold it down here, hold the air out, push the air
I do
out.
have a, I had
Yeah,
a scale and I did,
While
was
there's
it last year? I bought a pressure cooker
now,
just
that'll help
because I
prevent
wanted
some of
to
the- some of the-
sterilize mushroom bait.
Well, it's
It's hilarious.
it's
I'm
good
like,
that you have all that stuff. The big thing is, it's just like,
Yeah.
like if you're trying to grow from liquid culture,
I think
you're gonna
I'm-
have the longest
Yeah.
amount of workload
I know, I'm gonna try,
to know whether you succeeded or failed.
yeah, I know, I'm gonna try the agar thing.
Agar, you're gonna get an idea. Oh, this is a fail
Yeah.
right before you go to the next
So,
phase.
yeah,
But
so probably
otherwise, you're gonna go through a lot of phases
So, yeah.
before you
probably-
know if you were successful.
Yeah,
Well, yeah, I usually just use a pair of needle nose pliers,
Yeah.
but.
Yeah, but that's
I think I'll do that, because I need to check if
Doesn't have to
those
be
liquid
insane.
starter. Oh,
And this one, don't use this as a
still
good example.
good
because
enough
mine's
to
too loose.
use.
I
Yeah,
think it's like.
all it takes is one molecule
And yeah,
of
and I
something
ruined it.
to be. Yeah.
Okay, you ready, babe?
Yeah, so I'll do an agar workshop next month,
Oh, you need a scooper?
probably
Oh.
second week of February. I'll put it on video. And we'll try all the different agar strategies.
Oh, okay.
Do you have any?
Are you sure you don't want
Yeah, I don't think anyone joined the YouTube, but that's okay.
I recently got some of the material for AdGuard. It's like I've
Alright.
have so much to learn. I haven't even thought about using it yet.
Yeah, go ahead and make a couple bags. I mean, there's plenty. There's enough spawn. There's more spawn than we can ever use, and I have over 100 pounds of substrate. So if we don't make bags with it today, it's going to go home and sit in my garage until it gets molded. Or I'll have to make a lot of bags for myself.
10 pounds.
Like ranking league.
It took me a long time to actually get started with agar. I bought all the materials and I just kind of looked
Probably
at
want to
it.
use that whole bit of brick here.
The best thing you can do is experience it once and go through the process once with help in a workshop environment. Then you'll be able to have more confidence when you do it at home.
Yeah.
So,
There's a lot of it's a big thing. There's a lot of variables.
a lot of variables, and yeah, it seems like there's like so many different ways you can screw it up, and that's not wrong. There are so many ways to screw it up, but it's not as scary as it sounds.
Yeah, failing is part of the process with this stuff. It's just choosing where you have the most risk in the fail. You know, like
One more thing.
you don't want to put all your eggs in one basket.
Because I'm like, I just.
They're expecting to fail sometimes. They want to like kind of spread it out.
Yeah, exactly.
Oh, what's the time?
Training and failing and failing, and then I bought like
That's a
something more
rest.
serious.
Yeah,
I
that,
bought
that's
like a
good.
throw bag with a turn, and I was going to use like I was doing the open where you so we did that.
So this is what the melee looks like.
What's that?
This is what the melee game looks like.
Yeah, so this is the green spawn. So the millet.
Then you can try to put your
Is that what it? I'm
Yeah.
not familiar with the term.
Oh yeah, that was my question.
Oh, okay, I'll have to write the JJ.
Yeah, we're breaking it up so that each one of those little morsels of millet can be as closely equitable throughout the whole medium of wood. So that if they're equidistant, then they'll, once the mycelium realize that infiltrated the whole domain of the bag, then they'll start to fruit.
And for all the information for this, I'll throw together like a quick slide and throw it on the meetup chat. I meant to do that today. But I went on some things today.
All right.
All right.
If you move that one down here, I'll move it since my hands are.
Can I throw the rest of that?
Oh,
I'll let you keep that. I'll let you keep mixing that. So,
Do you want to just next
and
time?
then just send your bag over there.
Yes.
Yeah. Then you just pack that and zip that stuff.
I could hold the, good keep, yeah, we want to get it done like
I like how often we use the alcohol. I was like, whoa.
Well, what's
But I
nice.
think, no, because like, just physically see it. It
Yeah.
helps to remember, like, okay, we need to keep it sterile.
Yeah.